Afternoon, y'all. Hope you guys are great. We are right here. It's the 9th. So we'll be reviewing some cell cycle control stuff, doing a couple of practice problems. Don't forget, next time I see you guys, the first dose of your ultimate review is due. So how much? Thank you. So how much stuff do I have to turn in for full credit? 4 pages, front, back, front, back. So I wanted to show you whenever it says, like up there at the top, it says like front, back of average size handwriting. You probably have a pretty good idea of what it is, but I grabbed some examples. So whenever I'm saying like normal, like that's pretty normal. So looks something like that. So that student, if that was front, back, front, if they had a 4th, this was for something different. This is not their ultimate review. So if they had a 4th page of that, I'd be like, cool, I can give that person 100. If I had something like this one, same thing, looks pretty normal. Cruising through, looks pretty word dense. So cool, I can count each of those. Obviously, that's like a half page right there. So all that's great. Got a diagram here or there. That's fine. So to me, it seems fair if I give that student 100, I would also give that student 100. Looks about the same. This one, a little bit more space, but college rule, paper slightly bigger. So all right, cool, that'll work, that'll work. All right, there's a little bit of space here and there. All right, cool. I can basically count each of these pages. That. There are less than 50 words on that page, so that won't count. That doesn't count. I'll count it, but I'll count that as like a 1/5 of a page or something. So if that student turns in, again, like that, I can't in good conscience give this student credit for a page if I'm giving this student credit for 1 page, right? Like that doesn't make any sense. So I'll be like, hey, here you go. You've got like a 1/3 of 100. And they're like, but I turned in 7 pages. I was like, no, you turned in like 400 words. So if you're unsure or if you're just like, I write big, cool, just write a little bit extra. Some of you all write really small. And so I've even given you stuff back and you're like, hey, this looks like it would count as 100. And I'm like, that's like 100 1/3. Like that's happened, right? Where because you write small, like you have to write less. So you've written like however much I've asked in the past, but because you write really small, I gave you like, instead of 1/100, I gave you like 100 1/2. And then some of you guys have even learned like, all right, so here's about how much I have to actually write for 100. So it scales the other way too. So you can write in your natural handwriting, but if you write like really, really big or you have like tons of space, then just make sure you write more. So like that, I can't call that a full page because that kind of rips off the other student. Sometimes I get like big diagrams. Hang on. See if I can find that one student. Oh, I guess I didn't grab it. Some students will write like really, really big diagrams that take up like half the page or whatever. So anyway, just make sure that you have average size handwriting. If you're to turn it in and it's not quite enough, I just be like, hey, give me another page and then I can give you 100. But that is due next time I see you. We're right here on the 9th, and so the next time I see you is the 13th. You'll eventually have to turn in another 4 pages before the AP Bio exam, so if you turn in like 8 pages next time I see you, you are done with all required homework in this class for the rest of the year. Okay, not a single smile. All right, you guys are sad. Don't forget, next week we have our 4th after-school review, and it'll be transcription, translation, gene regulation, all of that stuff. I think that's pretty much it for announcements. Any questions about schedules and all that kind of crap before we review some cell cycle? All right. We'll start off. I had a question for you. So give this one a shot. You can jot stuff down if you want. If you ever want to take a picture of these, that one's fine. Or, you know, it's cool. Go ahead. Okay, we'll give it maybe 20 more seconds or so. All right. So for this one, it says, A skin cell completes one round of the cell cycle. Describe the products. And you're kind of like, I don't know, describe the products. That could be something pretty big or something pretty small. This was a 4-point question, A, B, C, D. So A is worth 1 point. So I'm trying to answer enough to get a point. So I want to be thorough, but I don't have to write like paragraphs or anything. And so what could you say for describing the products of the cell cycle? What kind of things could you say? Huh? Yeah. So I've got 2 cells and they're identical. What else could I say? Huh? They each have their own nucleus and organelles. Yeah. They each have their own nucleus and organelles. Are they diploid or haploid if it's at the end of mitosis? They're diploid. Meiosis makes haploid cells. Mitosis makes diploid cells. If you go and you look at the little grading rubric, it basically says, like, if you say that you have 2 cells and they're identical or they're genetically the same or something like that, then you're good. So they have to somehow let us know that they're the same and that there's more than one. All right, so that was a point. Based on figure 1, explain how p53 regulates the cell cycle in the presence of damaged DNA. So if my DNA gets damaged, then how does p53 regulate the cell cycle? What could you guys say? Yeah, okay.
Like when the DNA is like damaged, so a signal like a production of kinases, like p53 is CDK. Kinases will like bind and bind the DNA and stop it.
Yeah. Here it's basically just saying, like, just sort of like explain this diagram, put this diagram into words. And so if I'm like, when DNA is damaged, it looks like that activates or turns on or whatever words I want to use. These guys, protein kinases, and then those guys, it looks like they activate p53, and then it looks like p53 inhibits CDK, and so that's like stopping the cell cycle or slowing it down or whatever negative word you want to use. So you're good. If I remember right, I'd have to click back through and look at it, but the bare minimum was something along the lines of when DNA is damaged, p53 is turned on or activated, or becomes busy, whatever word you want to use, and then that will inhibit CDK. So they were mainly looking at that, and that. All right, for C, where would I draw the X? What phase of the cell cycle? Yeah, the S phase. So I copy DNA in the S phase. So real quick reminder, what was it that I did in the G1 phase? Yeah, the cell grows. Yeah, it gets bigger. S, we copy DNA. It stands for synthesis, but that name doesn't really help me that much. Synthesize means to make stuff, but it's copying DNA, so making DNA, copying DNA, I don't know. What about G2? What do we do? Yeah, we've got to copy the other stuff in there, the organelles, and then in the M phase, we got mitosis and cytokinesis. That's what's kind of dumb. M phase includes both mitosis and cytokinesis. And the reason I find that dumb is, first of all, you think the M phase would just be mitosis. But second, the biggest complaint that scientists have is that, like, students will think that mitosis and cytokinesis are like one and the same. They don't really separate them. And then they're like, why don't they separate them? It's like, well, you didn't. That's why we mentally don't separate them. And then G0, we might have mentioned that briefly back in the day. What's G0? Yeah. There are checkpoints along the way. So it's not necessarily a checkpoint, because I've got checkpoints in like G1 and in the S phase and around here in M. What's happening? What's it trying to show right here? Yeah. Yeah, leaves the cell cycle. So sometimes cells quit dividing. Normally, cells are just in the business of always dividing. And so if they are, great. I might slow down a little bit, I might speed up or whatever. So some cells go through a cycle pretty quick. Cells that have a high turnover like your skin or maybe the inside lining of your digestive tract, that's a pretty rough environment. Can somebody give me an example of a cell that goes into G0 and basically stays there forever? Yeah, Helena. Yeah, neurons. So central nervous system neurons generally don't divide, so brain and spinal cord. Luckily, my peripheral nerves are usually able to divide. So I can get a cut and damage my arm, and then like the nerves can grow back. I'm not going to be like numb. Whereas brain and spinal cord nerve cells, as a general rule, the genes that allow them to divide originally, it's like they get shut off and they just seem to be like permanently locked in the off position. Yeah.
What about when there's scarring? Like on the peripheral nerves, like
when there's what?
When there's like scarring on the skin,
Mm-hmm.
what the peripheral nerves still like divide, or would they like not?
No, so scarring is whenever, if I have a small cut, like, I don't know, just like how many teeny tiny cuts have I had all over my arms from like moving from this house to that house, chopping wood, carrying, like you get tons of little bitty scrapes and you can't tell it all. But if I get a cut that's deep enough and my body can't repair it very well, then it puts in collagen, and that's a scar. So collagen is white, and that's why scars look kind of like white and silvery. Your ligaments and tendons are made mostly out of collagen. If you're ever like eating steak and there's that weird sheath on it that's like really, really tough and kind of shiny silvery, that's high in collagen. So if I have collagen in there, then like nothing is normal. So that's just like a big old blob of collagen. So the nerves aren't there, the skin's not there, the muscle's not there, it doesn't even look the same. It looks like a scar. So scarring, it doesn't. But if I damage peripheral nerves, they can grow back. Now again, with a scar, they don't. Whereas if I damage brain and spinal cord, as a rule, they can't. So at some point, we'll be able to turn those genes back on, because obviously my brain was growing when I was like developing as like a newborn baby, right? So they can divide, it's just the genes seem to be off. So at some point, we'll figure out how to turn them back on, and then maybe we can fix strokes. Yeah. Do what?
Telomeres?
So telomeres, why was it that I have those? Why do I, or where are they, first of all? If I was looking for telomeres, where would I go looking for them? Yeah, on the ends of DNA. And why does it make sense that they're on the ends of DNA? Why are they normally not really anywhere else in my DNA? Yeah, that's the part that shortens every time my cells divide in the S phase. Then I lose a little bit of DNA on the end, and so I just have some gibberish sequences at the end. In humans, that's usually TTAGGG. Sometimes it's TTGGGG, but it's just gibberish. And so as I lose them, it's no big deal. Telomeres determine how many times a cell can divide. Me exiting into like G0 or whatever isn't related to telomeres. So it's not that my brain cells couldn't divide anymore. It's just that the genes that allow them to do it for some reason are off. So they still have divisions that they could do. It's not that like I reached the end of my telomeres and so now I go into G0. So it's kind of weird. But basically, if we can take brain cells and take them out of G0 and push them back in there, then we ought to be able to like fix strokes. And like brain damage doesn't have to be permanent brain damage. That'd be kind of nice. So. All right, so our X would be anywhere on the S phase. And I think we had one more. Part D. Based on the figure, explain how a P53 mutation may lead to an increased risk of cancer. Anybody want to take a crack at that one? Especially if you don't normally talk. So you guys, it's April. I don't know if I've ever heard your voice in front of your peers. How does mutated p53 increase my risk of cancer? In fact, it's one of the best predictors of whether or not I'm going to have cancer. All right, I got it. So p53, whenever, in this example, my DNA is damaged. I guess I was out in the sun from UV light, or maybe I just, hang out with UV light. And so my DNA gets damaged, and then what it's supposed to do is stop the cell cycle from continuing. It's supposed to inhibit it. Well, now it doesn't. So p53, if I was trying to write that in a shorter way, I'd say something like p53 is supposed to stop the cell cycle when there's damage. If p53, the brakes, are broken, then the cell cycle continues. And cancer is cells growing out of control. So there you go. All right. This one isn't too bad because it's almost all biology content. There's not a whole lot of reading comprehension and trying to look at a diagram and look at 3 different graphs and keep it all in your head or something like that. So that's what you'll find. Some of you guys will feel more comfortable with the stuff that's just straight biology content. Some of you guys like the stuff that's more reading heavy. You get a little bit of all of it. All right, now we're going to review some cell cycle stuff. So I got some DNA. How much DNA do I have in a human cell about if I stretch it out? More than that. Yeah, like 6 feet. So a couple of meters if you're British. And so we got to take that DNA and we've got to get it to all fit into a cell. So how is it that I get DNA? Talk with somebody around you. How is it that I get DNA to fit into a cell without getting tangled? All right, somebody raise their hand and tell me what you think. How do I get that DNA into a cell without getting tangled so I don't have to die? I don't want to die. Don't let me die up here. Yeah. Yeah, wrap it around histones. Good. And then what part of DNA is attracted to histones? Yeah, the phosphates. And what about the phosphates attract to histones? The negatives. Yeah, right there on my phosphates. Anytime I think of phosphates, I'm always going to think about the fact that they are negative. So if somebody started talking about lipids, the first thing that I'm thinking of is I always think of lipids as being nonpolar. And ions or things like phosphates have charges. So histones are positive, phosphates are negative, so DNA will wrap around these histones. If I wanted DNA to let go of the histones, what can I add to the histones to get the DNA and the histones to let go of each other? Yeah, acetyl groups. And so those acetyl groups can kind of cover up the positives on the histones. So if I had some histones with positive charges all around them, and my negative DNA was attracted, then I can basically go in with acetyl groups and like cover those up, and now my DNA lets go of histones, and that's kind of one of the steps in turning genes on. So adding acetyl groups means I can turn genes on. What was it that I could add to promoters to turn genes off? What did you say? Yeah, methyl groups, so CH3. Now, you might wonder every once in a while, like, hey, I'm eating my cereal and I'm reading about vitamins or whatever, and you're like, hey, I wonder why I need this vitamin, or I wonder why I need that vitamin, or why is it that I'm supposed to have this nutrient? So the reason for some of them is they provide methyl groups. So one day, if you're a female and you think you might be potentially getting pregnant, you're married and you're like, all right, we might have kids in the next couple years. And so you can start taking prenatal vitamins. And so the difference between a vitamin and a prenatal vitamin is prenatal vitamins have extra of, oh my gosh, I forgot. No. It's one of the B's. Folate, also known as folic acid. And folate or folic acid is a source of methyl groups. So females that are pregnant that don't have enough methyl groups, when their babies are basically cooking up inside of them and it's time to turn off genes, if you don't have the methyl groups for it, you don't turn off those genes. One of the worst things that happens if you're running low on methyl groups during certain stages of pregnancy is spina bifida, and that's when your body fails to build a bony spine around your spinal cord. And if it's bad enough, your spinal cord, when you're born, is on the outside of your body, meaning like right now, you'd be touching my spinal cord. Well, damaging a spinal cord is easy. Some of you guys are in anatomy right now, and I think you're dissecting brains, right? With those brains, those of you guys that are dissecting them, you're probably not a freak. You probably didn't just go like, but you've been cutting on them and touching them. Wouldn't it be easy to just take that brain and just completely just like that? That's how easy it is to damage your spinal cord. So spina bifida is lethal, and it's all because mom didn't have enough methyl groups to turn genes off when they needed to be turned off during week 5 of pregnancy or whatever. So that's why you're supposed to have certain nutrients. That's not all of them, but some of them are like, hey, we need sources of methyl groups. All right, cool. So I wrap DNA around histones. I shrink it down and everything else. I can shrink it down and shrink it down and shrink it down more and more and more. And then eventually I can make chromosomes. They kind of have all these names for chromosomes, like that chromosome right there is one single strand of DNA, just like coiled and coiled and coiled. If I look at just the DNA, I call it DNA. If I look at the whole strip of DNA wrapped around histone proteins, I call it a chromosome. If I'm looking at like a chunk of that, they call it chromatin. Chromatin ends in IN. So what would I assume about it? Yeah, there's some protein in there. So chromatin is DNA wrapped around some histone proteins. It's not pure protein, it's DNA wrapped around protein though. So what was this picture? I'll give you a chance to talk with somebody around you. What's this thing called? A picture of my chromosomes. A photograph of my chromosomes. A microscopic photograph of my chromosomes. All right, what do you think? Somebody raise their hand and tell me what that thing is called. Just a vocab word. Yeah, a karyotype. So a karyotype is a picture of an individual's chromosomes. Good. A genome was like, what's the standard for that species? So as a human, I'm supposed to have 23 pairs of chromosomes. That's what's considered normal. And this person's karyotype has 23 pairs of chromosomes. Is it possible that my karyotype could have something different than 23 pairs as a human? Yeah. So what's the most common disorder that we think of that has a different number of chromosomes? Yeah, Down syndrome. So if I have a third chromosome 21, that's Down syndrome. There are other diseases where I have extra chromosomes or not enough, but the one that we hear about the most is Down syndrome, and that's because it's possible that I can be born with Down syndrome and survive. Whenever I'm like missing a chromosome 2, that's lethal. That's a miscarried baby. If I have a third chromosome 2, that's lethal. That's a miscarried baby. So the reason you never hear about trisomy 2 is like mom loses the baby at like week 6 or week 8 or something like that. So that's why you don't really hear about those. Why is it that I understand like why maybe like missing a chromosome is bad, but like if I had a third chromosome 8, it seems like I have a backup chromosome 8. Why is that bad? Why is that lethal? I totally understand when I'm missing stuff, that's bad. But like having an extra, almost like I'm supposed to have my math book, but I've got 2, and so I don't know. Yeah. Yeah, I make extra of everything. So when I'm supposed to have like, 2-letter V's to make something, well, now I've got 3 of them. So now I make 50% more of whatever that stuff is. So when it's time to go through and make hemoglobin, it might not be a big deal. It might actually be a bonus. But I would read, let's say my hemoglobin gene is right there, and then there's one right there. So I would read that, I'd make hemoglobin. I'd read that, I'd make hemoglobin. If I had a third one, I'd make a third hemoglobin. That might be totally fine. Some stuff I can make extra of or less, and it doesn't matter that much. But some things, it's catastrophic. So you're messing with the ratios of proteins. So I always compare it to some sort of recipe. If I'm supposed to have 2 cups of something, and instead I only put in 1 cup, I probably screwed up the recipe. If I'm supposed to have 2 cups and I put in an extra, a 3rd cup, I probably screwed up the recipe, right? So that's kind of how I think of it is I'm supposed to have 2 cups of everything. And if that's wrong, it might be bad. For one protein, it might be better. Extra cinnamon in the cinnamon rolls, I'd call that a plus. But there are some things that it's going to be bad. So extra brown sugar, I mean, come on. It's good stuff. All right. Now, let's look at some cell cycle stuff. All right, we just went through. There we go. We just went through like all these different phases right here. Now we're going to focus on mitosis. So take maybe 2 minutes, jot it down, or talk with somebody around you. What are the phases of mitosis, and what happens in each? All right. So in AP Bio, you can break it down into 4 or 5 stages. Prophase, this very last one you can just call more prophase, or you can call it prometaphase. Metaphase, anaphase, and telophase. So PMAT, and then that one right there is, PM. So in prophase and prometaphase, I got 3 things that happen. Can somebody give me one of these 3 things? Yeah, Caden. Yeah, so I've got to have centrosomes form, yeah. So centrosomes appear, and they make spindles or spindle fibers. So I need centrosomes to appear because they're the things that are going to make the spindle fibers to like pull stuff apart. Yeah. Yeah. The last thing that happens at the end of prophase or kind of prometaphase is the nucleus breaks down or dissolves or whatever word you want to use. Some enzymes are going to take care of it, but it'll go away. Uh-huh. Yeah, I got to pack down my DNA. It's all kind of strung about, and so I've got to pack it down into chromosomes so it's easier to move around. So my DNA will like condense into chromosomes. And the idea behind that was if I've got big old long strings of DNA and I want to move them somewhere, if I just kind of like grab it here and then have to move it, it would be hard. So imagine this was hundreds of feet long. If I grab the end and I move some of it here, most of it's still spread throughout the room. But if I can like take all that DNA and like spool it around histones and pack it down into chromosomes, then it's easy to move if it's all like spooled together. So, all right, so I condense my DNA so it'll be easier to move. Centrosomes appear and they're going to make spindles to grab the chromosomes and pull them apart, but the nucleus was in the way. So you might remember in our pictures of this that my centrosomes are made on the outside of the nucleus. My chromosomes are inside the nucleus, so those spindles are trying to grab the chromosomes, but the nucleus, or the nuclear envelope, if you want to be fancy, is in the way, so my nucleus has to break down. All right, how would I know that I'm in metaphase? If I'm, yeah, so my chromosomes are lined up in the middle, I think a middle metaphase. And then what's the major thing that happens in anaphase? Yeah, apart. I pull them apart. So pull chromosomes apart. So middle metaphase, A apart. And then telophase, if I'm writing for the AP Bio test, again, I'm going to list all three things. But just for me mentally, what are those three things? Yeah, opposite of prophase. So I would go through and I'd literally just say the opposite of each of these. That way I can get like 3 points. If I say opposite of prophase, and that's because we only did prophase to prepare to divide, and now we're done dividing my DNA, so we put it all back like we found it, it's like, cool, we'll give you a point. But if I can like list out each of the three things, then now maybe I can rack up like 3 points. So always remember, anytime that you know something that you either learned in this class or learned in like you remember from Biology 1 or whatever, always write it down. Otherwise, we can't give you points for it. So the classic example that's just easy for you guys to grasp is like chi-square. If you know the p-value I'm looking for is 0.05, then I can't give you credit for that if you don't tell me what it is. Being able to read that chart, that's a skill that you didn't know until you took stats or this class. So, hey, when I'm reading that chart, can you give me any information? Like, hey, my critical value that I'm looking at, according to the chart, is 5.991, or there's this many degrees of freedom, and here's how I got that. That's like knowledge. So anytime that you can give me a piece of knowledge, that's potentially a point. All right, cool. So I'm cruising through the cell cycle, and there we go. There's the rest of it. Once I'm done with telophase, then the cell actually splits. Cytokinesis translates as cell moving. So the word is like sort of useful, but I'm splitting the cell into. A lot of times teachers will say like, in mitosis I'm dividing the nucleus, and in cytokinesis I'm dividing the cell, or something like that. So you can think of it like that. Some teachers have like little hand motions they do in like Bio 1 where they're like, you know, this is prophase, this is metaphase, everything's lined up in the middle, anaphase, 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 and then like telophase. It's like prophase, metaphase, anaphase, and then I like rebuild the nuclei and stuff. Telophase. In telophase, I uncondense my DNA, centrosomes and spindles go away, and then now instead of the nucleus breaking down, I like start building new nuclei. So prophase. Metaphase is supposed to be them like lined up in the middle there. Anaphase and telophase. Huh? It depends. So in human cells, it would take longer than you think. So most of the time when your cells are dividing really, really fast, like as you're developing inside a mom, it's 8 to 12 hours to run a cell cycle. Now that you're born, fast is like every 24 hours. So like if I get a cut and I'm trying to like repair that cut as fast as I can, my cells are probably still only dividing about once every 24 hours. So compared to how fast all kinds of other crap happens in my cells, it just, I don't know, I would have thought it was faster. Yeah.
What about like just mitosis?
Just mitosis? I don't know. I know that normally, I feel like I have that in my notes. No, I'm mistaken. Mitosis takes 24 hours. And then G1, S, and G2 are highly variable from cell to cell. So everything I said was not cell cycle, it was mitosis, so I misspoke. Yeah. Anyway, so it is surprising, but at the same time, when I'm doubling every single time, then cells can grow pretty quick. So by the time I've got 100 cells, and then it's 200 cells, 400 cells, the way that I can patch up this cut in, say, 48 hours or 3 days or something is if I have thousands and thousands of cells that are all dividing at the same time. then I can kind of fill the gap a little bit quicker. But yeah, it's slower than I would have thought compared to all the other crap that seems to happen in our cells so fast. We got checkpoints along the way. They love talking about checkpoints. In the after school review, we talked about checkpoints briefly, so I'll be kind of brief here. But we've got checkpoints along the way. And a checkpoint, as far as like what happens at each one, I just need to use logic as a general rule. If I'm near the end of some sort of phase, then the checkpoint is just checking to see if enough of that thing happened. So in G1, the cell is supposed to grow. So that checkpoint is just checking, like, is the cell big enough? If it is, you can keep on going. So I can just sort of like kind of guess what would I be checking for here. Like, does my DNA seem to be copied? Does it seem like I've copied organelles? Cool, move on. So checkpoint is checking your work. All right. This part will be repetitive from the after school review, but it is important, so real quick. If I want to go from one phase to the next, I've got these things called cyclins, IN. They're proteins that allow me to continue through the cell cycle, cyclins. They're always around, but their default position is off. How do I turn on a cyclin? What do they depend on to turn them on? I'm sorry, CDKs are always around. I misspoke. CDKs are always around. They depend on cyclins to turn them on. So CDKs are always around, and if I want to activate them to go to the next phase, then I start making cyclins for that. So for example, these are the ones that are allowing me to go from G1 onto the S phase. So I've got these CDKs and they're off. As my cell gets bigger, somehow, again, we don't know how consciousness works, somehow it knows as my cell gets bigger, make more and more cyclin D, make more and more cyclin E, and I will. And so as the cell gets bigger, it makes more of these cyclins, which means it turns on more of these CDKs, which means it says proceed. Cool. All right, now, maybe the cell is big enough to move on, but that doesn't mean that everything is cool. So just because my cell is large enough doesn't mean that there's not like problems because I was exposed to UV light or something like that. Or the example that's the easiest to understand, I've got some DNA damage or something. So even though my cell's big enough, if my DNA is damaged, I want to like stop the cell cycle. So take a moment, talk to people around you. Out of all of these proteins out here, there were two main categories. So what were those? Some of you guys heard this 2 days ago in the after school review. All right, somebody give me one of them. We can classify all these proteins as one of these two things generally. Yeah, proto-oncogene. And then what was the other guy called? Yeah, a tumor suppressor. And proto-oncogenes are any proteins that their final effect is telling the cell keep on dividing. Proto-oncogene, onco is cancer, so cells dividing. Onco is cells dividing out of control, so that makes sense. And then tumor suppressor, when cells are dividing out of control, that's cancer. That results in tumors. So tumor suppressor, it makes sense that those would be stopping cell growth. So talk with somebody around you. What about AKT? Is AKT a proto-oncogene or a tumor suppressor? AKT, proto-oncogene or tumor suppressor? Oh yeah, it's totally fine. It's still totally just effort-based. So if you're skipping around, if you tend to do like chunks of biochem or something, if you'll just put like biochem and then those, and then if you like switch to cells, put like cells or like kind of these little mini titles along the way. But yeah, if you skip around, it's cool. It's still mainly effort-based. I'm just like, yeah. Half the time I don't even know what question you're answering. I'm just like, oh, that's true. Oh, I bet they're talking about protein structure. Oh yeah, they're describing inhibitors or whatever. So yeah, it's cool. All right, so AKT, we'll vote. Raise your hand if you think that AKT is a proto-oncogene. A few for that. Raise your hand if you think AKT is a tumor suppressor. It's like a horrible voter turnout right there. It's like we're voting for mayor or something. Or Blaylock. I don't know who Blaylock is, but I feel like that dude's had signs up for like my entire life. Alan Blaylock? I don't know what that is, but like I feel like he's been running since like 1998 when I moved down here. Anyway, so the way that I look at it is I can do the easiest way is the plus minus method. It's a proto-oncogene, by the way. So AKT, that is stopping that one. So I think of that as a minus. So I have a minus. And then that causes Fox to be that guy. Do you see why I'm calling this a plus and that one a minus? That one's saying stop and that one's saying go. So to me, it makes sense that plus means go and minus means stop. That's a minus, that's a plus, and that's a minus. So in math, a minus times a plus times a minus, that's a positive number. So it's saying go. It's a proto-oncogene. Makes sense. Right here, P16, that one's easy. That one's saying stop. And that's a minus, right? This one, okay, P18, that one's saying stop. Isn't this one activating? Pretend that this is a protein. Isn't this activating this guy? So is he telling him to say stop? So plus minus still means stop. This one right here, BMI1, isn't he stopping this guy from stopping? So he's stopping this guy from hitting the brakes. This guy's the brakes, and he's stopping him from hitting the brakes. If I'm the brakes and you stop me from doing my job, then we keep going. So a minus and a minus is a plus. So you might be like, well, hey, Mr. Cotton, what if I went this way? It's going to give you the same result. That's a minus, that's a plus, that's a minus, so that's still a proto-oncogene if I follow this pathway. If I go this way, that's a minus, that's a plus, that's a minus, so it's still a proto-oncogene because AKT is always a proto-oncogene in this cell. In some other cell, it might run a totally different pathway, but in this diagram, if I start tracing stuff, you're not going to find one pathway says go and one pathway says stop. If it does, there has to be a way to like only one pathway is on, and you know this time this pathway's on, and this time this pathway's on, or something like that. So that's how I think of them. Is the plus minus method is the easiest. All right, cool. Let's say that I damage my DNA. The cell somehow realizes I've damaged my DNA, and it knows it can't repair it. Talk with somebody around you. It's a vocab word. What should my cell do? I'm a cell. My DNA has irreparable damage. What should I do? Irreparably damaged, can't be repaired. What should I do? All right, what was that process called? What are we hoping to start up? Yeah, apoptosis. So apoptosis is programmed cell death. So we think that all living things that we've looked at, we haven't found an exception yet. Kind of like we think that everybody has unique fingerprints because we've never found two people with matching ones. We don't have everybody's fingerprints in the world on file. Your fingerprints likely, when you guys were born, you guys might have been fingerprinted when you were born. Starting with somewhere around, I can't remember if my son was fingerprinted or not. I was super tired. Anyway, but people my age weren't fingerprinted when we were born. They would footprint you. So we have footprints of everybody that's my age, but we don't have fingerprints of everybody that's my age. And you might go, why? I don't know. Why does Starbucks cost $6? Right? Why does Radio Shack ask for my phone number when I want to buy a pack of batteries? I don't know. That's a Seinfeld reference back when Radio Shack existed. Okay. Anyway, so apoptosis. So I've got genes that say cell destroy yourself, but normally they're off. If a cell gets damaged enough, then we'll turn those genes on. And so in general, the reason that your cells are still alive is you have this combination of gene pathways, the red ones that say destroy yourself. Most of you guys can't read this from where you're at, but we saw this way earlier in the year. I have death receptor, and it links up with death ligand, and those are all telling the cell to destroy itself. Over here, the green ones have growth factor receptor, and they are activated by growth factors. So the reason that my cells are staying alive right now is I have the right balance of these to where the cell isn't destroying itself. If something seems to go super wrong and somehow my body can figure it out, my cells can figure it out, then all of a sudden it will turn on the apoptosis genes. If I'm a white blood cell and I bump into a cell and I don't recognize it, I can send chemicals across that turn on these apoptosis genes. I can send across death ligands and tell the cell, destroy yourself. And it does. So it's just, you walk up, you're a white blood cell, you touch this cell and you're like, kill yourself. And the cell is like, all right. And it destroys itself. Just one cell, not the whole person. No reason to get upset. Yeah.
I know that like cat, I don't know what it is, caspase.
Oh, caspases?
Yeah, it says dependent and then independent. I know in the video that like the animation.
Yeah.
It was independent.
So they kind of show dependent and independent. Essentially what it comes down to is there's more than one pathway. It could be externally, I could be told by another cell to destroy myself, or I could decide on my own to destroy myself. That's chemically, it gets more complicated than that, but like as far as like conceptually what's the difference? It's, do I decide on my own, I messed up, I should destroy myself, or does a white blood cell have to say like, hey bud, you should destroy yourself, you got cancer, something like that. So it's almost like an external signal that tells me to, or an internal signal that tells me to. So I won't make any parallels with humans because you guys didn't like that. It looks like this. So remember, when apoptosis happens, it's pretty intense. The cells just rip apart their cytoskeleton, and then they bleb out. All right, let's see. Sometimes apoptosis, even though it always looks like that's got to be bad or something like that, sometimes apoptosis is the plan all along. So there are cells that are destined to destroy themselves. It's almost like you've done your job, now we throw you away. It's like a candy wrapper. Like, it had a function. Once I eat the candy bar, its function is over, so like, you're trash now. So whenever you're developing limbs, you had webbing that went like all the way out to the edges of your fingers, so your feet and your hands looked more like kind of duck feet. And it was destined all along that most of those cells were going to destroy themselves, so that you don't have webbing like all the way out to your fingertips. And so this, it's obviously colored, but it's showing these genes turning on that are making apoptosis happen. So it is possible that apoptosis isn't always because some cell is sick. It's not always, I've got cancer, I should destroy myself. Sometimes it's like that's the plan all along, is the webbing in my hand, I'm a human, so I'm going to destroy myself. Or I'm a cardinal, so I don't keep duck feet. I have cardinal or robin or crow feet or whatever, like that. The other cancer stuff should be good on that. They do, not that you would have had to have seen these pictures or anything like that, but we can label stuff with like fluorescent proteins. And so it's very possible, they have questions a lot where they'll be talking about some experiment and they label something with like a green fluorescent protein or a red fluorescent protein, and that's how we watch it and all that. Just realize that that just means I can see it now. So the green fluorescent protein just glows green. So as they're explaining something, if they're talking about it, that just means like, hey, I can see it. Sometimes we radioactively label stuff. So if they start talking about like radioactively labeled phosphorus in DNA, and you're like, what's that? And you're like, well, the P's and the phosphates, they're radioactive. So now I can like trace them whenever they go somewhere. I can see them. So it's just a way of kind of tracking stuff. More cancer stuff. Don't smoke. Don't drink. Don't be born with Proteus syndrome. I'm not saying don't be born. It's probably an incorrect pause there. See, my English teacher was right. Like, do you put a comma or not? Sometimes it matters. Let's see. If I was comparing mitosis to meiosis, take a moment, talk with somebody around you. What are some of the differences? You ought to be able to come up with like 2 or 3 kind of big differences between mitosis, cells dividing, and meiosis, cell division.
You have it goes from one cell to one other so they make oh, it is. Yeah.
I hear you guys over there making fun of me. Don't be born. Now you can use that. You can use that with your friends. This is called Proteus syndrome, and so by some bizarre, it's what Elephant Man had. This is Elephant Man. And Proteus syndrome is not very well understood because it's super rare. It's been forever since I read this, probably 20 years. But at the time, there was only, on average, about 50 known cases of Proteus syndrome worldwide. So because so few people have it, like, we don't really know, what do people with Proteus all have in common? And you're like, I don't know. Like 2 dudes in the U.S. that have it. So, like, we don't have enough data to know, like, this is common in people with Proteus. And so for some bizarre reason, I don't know why, it doesn't qualify as cancer. That looks like cells growing out of control. But Proteus syndrome, there's several other things that look kind of similar. There's pictures of the Elephant Man. His name was like John Merrick or Joseph Merrick. And so oddly enough, Proteus syndrome seems to hit mainly one side of your body and not the other. It's not always the right, it's not always the left, but it's usually one or the other. And you can see that in him. It's not perfect 50-50, but like that part looks normal. Outside of the rib cage, like his arm looks normal, his chest looks normal compared to like that side. None of that looks normal. His hand looks normal. That does not look normal. So we don't know why. Because again, very few people have it. It's genetic, so you don't catch it, we don't think. And the way that they diagnose it is by looking at your big toe. The giveaway, the thing that they found that most people with Proteus syndrome have in common compared to other things that look like disfigurements is the way that their big toe looks compared to the rest of their foot. The big toe kind of sticks out in like a certain way. It's a little bit like if I was trying to describe to you without showing you a picture of somebody with Down syndrome, there's just certain characteristics that if you look at 30 people with Down syndrome, and then I show you 2 others and I ask you, hey, do these people have Down syndrome? You guys would probably be 100% accurate. You'd be like, he does, he doesn't, he does, he does, right? There's just certain characteristics. It's not just that I have a round face. It's not just that I have like kind of a flatter face or something like that, but there's sort of like you've got enough of all these traits. You know what I mean? So with Proteus syndrome, again, about the only thing that they found is there's a certain look that your toe has, your big toe, the way it sticks out. It's a little bit more separated from the others, but a lot of us have a big toe that's got kind of like a bigger gap in there, almost like normal people have like flip-flops, and they have something like that in there. And some of you guys are kind of like freaks. You got flip-flops, and you can fit like that in there or something like that. Proteus is like that. So you could have like a belt for like your flip-flop divider, like a big old like. So anyway, it's super strange. There were several movies about Elephant Man. That's remodeling, so that's not real. That's like computer generated. But that's either his real. Got a picture from, that's him. So King's College is where his bones are stored. So this is an actual photograph from the museum from multiple angles. So that like is his, that's like if you go to the Louvre and you're looking at the Mona Lisa, you're like, that's the real Mona Lisa. So they tell us. So anyway, yeah. It's horrible. If he laid on his back, he couldn't breathe. And so he died in his 20s. They found him on his back, and they don't know if it was on purpose or not. So it's a very sad story. So if you watch any of the movies about Elephant Man, you're like, this has a happy ending, doesn't it? And you're like, no, it just gets worse and worse and worse. And then they roll the credits, and you're like, oh my gosh. Got to go watch Forrest Gump or something now. Anyway, I guess that, yeah, don't watch that either. That's also. All right. So, okay. So the last thing that we want to do is try a free response. Now that you're good and fatigued, it's the end of the day. Bam. So back in 2023, question number 1 was this. Now I've got paper copies for you. So let me hand that out. 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6, 7. There you go. There you go. There you go. You're welcome. There you go. All right. I'll give you guys a little bit more light. I know some of you guys won't like it, but you'll be okay. All right. So if you're watching the video and you don't want to try to stare at a screen and read it, if you Google AP Biology Free Response 2023, number 1, this will pop up. So the reason I'm showing you this one is you'll do 90 minutes of multiple choice. You get a 10-minute break where you'll go and like eat a granola bar and chug a Coke or something. And then you'll come into the free response. You'll be like, all right, I'm ready to go. And then they're just like, ha. So question 1 seems to always look brutal. So there's a full page, and they haven't even asked me anything yet. Then there's a data table, and then they finally start asking me stuff. So question 1 is going to be worth between 8 and 10 points. So I'll give you several minutes. Probably not enough time to get through all of it, but at least get through part of it. I want you to try this first for me, though. This is an exercise, and yeah, it's a good exercise. Don't even bother reading all the background yet. See how far on A, B, and C you can get without even like, again, I might have to reference this, but instead of sitting down and like reading that paragraph and like mentally understanding these diagrams and then reading that paragraph, just see, bless you, just see how much can I answer without really reading much of it at all. Like just reading little bitty bits. Maybe I have to go back and like find some number, but I'm not like really digesting it all. So see how far you can get on like A, B, C, D without reading it all, without reading the background stuff. We'll hit pause on you guys. All right. So we won't look at all of them. We'll go through like A and B, and then I'll pause and let you keep on going. I did find what I was talking about with that Proteus picture. So obviously, if one of my feet is like super disfigured, where like if the whole side of his body looked like that, but apparently there are other diseases that kind of look like that. And so if you're trying to diagnose Proteus earlier, kind of like by the time somebody's super old, figuring out it's like, oh, they've got this disease, and you're like, he's 37 years old. Like if you could have diagnosed that when he was like 2, we could have done something about it. So as far as diagnosing Proteus early, there's just kind of weird thing that tends to happen with the toes. They'll call it toe hypertrophy, but it's not just big toes. It just, again, it just has like an appearance. All right, so the point that I was trying to make on number 1 is finishing the free response on time is difficult. It's harder than finishing the multiple choice on time. So if you struggle to finish the multiple choice on time, you will definitely struggle to finish the free response on time. The free response is written, so there is like a little bit of stamina involved. It's not typed. There's a little bit of stamina involved in like writing. If your handwriting is really bad, remember, they don't grade your actual physical paper. They grade a scan of your paper. So it's almost like whatever you write, imagine I go and I throw it on a Xerox copier, and then that's what you're grading. So if you write like super, super, super light, they aren't going to be able to see anything. That's why they're going to encourage you to write in like blue or black pen. Some people are like, I wrote in pencil, I bet it's fine. It's probably not fine. If you have a dark number 2 pencil, it's probably okay. But in general, what I find is this just kind of a life lesson. I can always ask why afterwards, but if somebody's ever like, hey, make sure you do this, and you're like, oh, I bet it's fine if I do that. Well, no, like, the mechanic said do this, like, and you didn't do it, and that's why this is messed up. So I would bring, like, a pen that you like. So you've got those pens that are not, like, non-globby pens, you know what I mean? So bring a blue or black pen that you like. For me personally, I'd write in pen. When it came to math, if it was, like, math I was pretty confident of, I'd probably write it in pen. If I'm like graphing and stuff, I can do it in pencil, and then I can always go back and trace it with a pen if I want. But you can have different colored pencils and pens and stuff, but just realize they're going to scan it. So the colors won't come through. So if I'm going to draw a graph, and I'm going to use like a black pen to show it's the one that had a high phosphorus environment or something like that, and then I'm going to use like dark blue. There we go. And I'm going to use dark blue for the other one. When they scan it, they're probably going to both look the same, right? So I want to label it. You know, this is like the high PI, and this is like the low PI. They aren't super picky on your procedures for labeling things. Like, do I have a little legend or a key or whatever you want to call it? It's mainly just like, do I know what this line represents? Oh, I do. Oh, do I know what that line represents? Okay, I do. Cool. So they're mainly trying to give you points for knowing stuff. Okay, so the point of this one is question 1 always looks bad. At some point, I can show you, you know, or you can just trust me. Question 1 usually looks bad. If you're cruising along and you see this and you're just like, oh, faux 80, faux 85, faux 4, you're just like, are you kidding me? Faux 1. In that data table, like, data tables are never intimidating, but like, if they were, it'd be that one. I mean, the title of this is just relative amounts of faux 1 mRNA in high PI environment plus or minus 2 standard errors of the mean. Like, come on. So it looks bad. However, pretend that I don't really read much of any of this, and I'm just like, all right, what's A ask? Describe the effect that the addition of charged phosphate groups can have on a protein that would cause the protein to become inactive. That's bold, so that's probably a point. Explain blah, 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 blah. That's bold, so that's probably a point. So if I add a charged phosphate group, what effect could that have on a protein that could make it become inactive? Yep, it'll change its charge. And so what would that do? It could repel it away from DNA. Would it possibly change the shape of the protein if I start changing charges? Like right now, if I have a chain of amino acids. Then if I'm building my primary structure and all these guys in the beginning are positive and all the guys right after that are negative, then won't those like fold on each other? Yeah. And then if this was nonpolar and this was polar, then they would like avoid each other. So positives and negatives are like what cause, and nonpolar and polar and all that, it's what causes a protein to fold the way that it does. So if I go in there and I start adding negatives somewhere, it might now like change shape, right? So that's what they're getting at. The thing that got me credit was if I start changing charges, that can change the shape of a protein. Explain how a signal can be amplified during signal transduction. So what is amplification during signal transduction? What do you think? Yeah.
Like more proteins are getting like the ball side, or like one, and then two signals are noticing more signals.
Yeah. During signal transduction, a protein can activate multiple proteins, and each of those can activate multiple proteins. It is okay to draw diagrams. Again, you're not typing your answers on a computer. You've got a pen, so you can draw little diagrams. Anytime that you're writing words and you're like, I know what I'm trying to say, but I'm not sure if the reader totally gets it, cool. Just draw me a little picture, and between your words and the picture, then maybe I can understand what you're trying to say. So make sure you kind of show me, like, how does this relate to this? You know, like, one protein activates multiple proteins, like the pic below. And then you might even tell me, like, that's a protein, or something like that, or activating many proteins, something like that. So you can draw little diagrams. All right, so that's a point. Did I have to know what was going on with Pho-8 and Pho-4 and all the other Pho crap? No, bless you. Next up, part B. Based on the table, identify a dependent variable. So a dependent variable is anything that I'm measuring. So I could pick that one, or that one, or that one, or that one. So I could barf out any one of these four things. When I looked at the rubric, what they needed was I would copy down all of it just to be safe. But what they needed, they didn't need the unit. So if you said that, or that, you got credit. So remember, the independent variable is the thing that I'm testing, the thing that I'm changing. A dependent variable is like what I measure. So over here, I know I'm measuring this. I've got 0.5 of it, of APase activity. Over here, I've got 17.3 of what I'm measuring, which is APase activity. Uh-huh.
Would it be like APase activity, and then, or APase activity, and then like how much
like all I have to do is like write that title. Like, so my dependent variable is literally just what am I measuring. So normally it'd be like whatever's over here. So like maybe if I was in a physics class, I'm measuring distance over time, then my dependent variable is distance. So I don't even have to know what relative amounts of Pho-1 mRNA in a low pi environment is, but like that's what I'm going to write. So it's just the title. I don't need any numbers. I'm either going to be testing it on wild type, mutant 81, or mutant 4. So these would be my, if they had asked me for my independent variables, if they're like, name an independent variable, I'd be like, all right, one of these yeast strains, and I'd pick one. Or if I said one of the yeast strains, they'd probably give me credit. Yeah?
Sorry, what are you going to write if you, like, this, like, APase activity and the PHR, or PHO, one out, or you have to do, like, a specific column?
Any one of the columns would work, because they're just asking for identify a dependent variable. We got 4 options. So that one, or that one, or that one, or that one. Any one of the 4, yeah. The environment. What do you mean the environment? It depends. To be safe, like if I say APase activity, some years, like I can look at this year, but some years they're picky and some years they're not. They're always consistent. So they'll decide, let's say I've got like 5 points available on a question. This one's easy to grade. This one's easy to grade. All of a sudden, once people start answering it, they're like, crap, this one's going to be really hard to grade. All right, are we going to be lenient or are we going to be tough? And they might decide to be tough. They'll be consistently tough on everybody, but they're going to be consistent. So a for sure right answer would be I write every single letter of that, down to the 600, the X with the line over it, all of it. If I put APACE activity, I might be all right. But the sure thing is I'm putting everything in that column. Do what? They often don't require it, but again, if I write all of that, it is impossible for you to argue that I'm wrong. The less and less that I write, the more you can argue that my answer is incomplete. So I'm going to write all of it. But you're probably okay. This year you were okay without the units. All right, next up, justify the researchers using the wild type strain for the creation of mutant strains, and justify the researchers using mutant strains in which only a single component of the pathway was mutated. So these two together, I kind of answer them together. I'm like, all right, well, I only want to have a single component mutated because that way if something's different, I know that that change is probably the cause. So if FO81 behaves differently from the wild type, then I'm pretty sure it was due to the FO81 mutation. So if I only have one mutation, then the outcome is probably due to that mutation. Why do I use the wild type strain to create the mutant strains? Well, now there's only one difference. The only difference between wild type and this is mutant FO81, or the only difference between wild type and that is mutant FO4. So it's a point of comparison. The thing that I'm trying to get across, because the bell's going to cut us short, wasn't I able to answer 5 points and I didn't even read all those paragraphs of crap? Now, is it helpful if I read them? Yeah. If I read those paragraphs, I would have a little bit more of an idea of why they were asking me about phosphate groups. They mention it right in there, so it would help some. But like, I can answer that without doing anything else. Identify the yeast strain that led to the highest relative amount of faux mRNA. And you're like, all right, faux mRNA, who had the most? 10 is the most. So wild type in the low PI environment. So I'm 6 points in and I didn't even read the stuff that they gave me. That's the point I'm trying to make. So we'll do some more things like this, but we're out of time.